A mammalian embryo faces one enormous problem. It is sealed inside its mother's body. It has no mouth, no lungs, no kidneys. And yet for weeks or months it has to eat, breathe and excrete. Evolution's solution is one of the most remarkable organ in all of the biology is the placenta. It is the only organ built by two individuals at once. Part of it comes from the fetus and other part comes from the mother. But here's what makes it fascinating for a zoologist. The placenta is not one thing. Across mammal, it varies enormously. In which membrane build it, how many tissue layers separate mothers from the fetus, whether it bleeds at birth, and how its villi are arranged. And that variations is exactly what your exam will ask you to classify. So today we will define it properly, then work through all four classification criteria systematically and finish with its functions. So let's begin. To obtain nourishment, the embryo develops a connection with the maternal uterine wall and its connection is called as the placenta. Functionally, the placenta serves as a passage for food and oxygen from the mother's uterine wall into the embryo and eliminates excretory products from the embryo into the body of the mother. It is a two-way street. So let's break that definition apart. So extraembryonic membrane. There are four membranes that forms outside the embryo proper. The amnion, chorion, allantois and the yolk sac. The fetal half of the placenta is always built from one or more of these. Apposition. The two tissues simply press up against each other without merging. And fusion. The tissue actually intermingle and invade. That single distinction, apposition versus fusion, is the seed of the entire classification we are about to build. Some mammals only ever oppose, other invade deeply, and how deeply the fetus tissue invades determines almost everything else about the placenta. There are four criteria. The first criteria is the fetal membrane involved. The second one is the histology, how many tissue layers separates the two blood streams. Third is the fate of uterine endometrium at parturition. The fourth is distribution of the villi. So let's talk about the first criteria, which is types of placenta according to fetal membrane involved. The question here is simple. Which extraembryonic material team up with the chorion to build the placenta? Remember, the chorion is always the outermost membrane. It is the layer that actually touches the uterine wall. So it participates in every placenta. What varies is which membrane comes up behind the chorion to supply it with blood vessels. Because a membrane without blood vessels cannot exchange anything, it needs a vascular pattern. So let's talk about yolk sac placenta. This is the primitive type of placenta. Here the placenta is formed by the contribution of the chorion and the yolk sac. The yolk sac grows out and presses its blood vessels against the chorion. It is found in some of the marsupials, for example the kangaroo and the possum. The next we'll talk about chorionic placenta. In this case, the placenta is formed by the contribution of chorion only. The chorion opposes the uterine wall without a yolk sac or allantois underlying it. It is the simplest and least effective arrangement, which is seen transiently in some marsupials. The third one we are going to talk about is allantoic placenta. This is the advanced and most effective type. The placenta is formed by the contribution of the allantois and the chorion. The allantois, originally an embryonic waste sac, grows out, fuses with the chorion and delivers a rich supply of blood vessels on it. It is seen in eutherians, placental mammals, and in some marsupials as well. For example, paramales and bandicoot, which is the classic exemption among marsupials. You will ask, where do human fits? Human placenta is chorioallantoic placenta. In humans, the allantois itself stays small and vestigial, but the allantois mesoderm supplies the umbilical blood vessels that vascularize the chorion. So by origin, it belongs firmly in the allantoic category A short-lived choriovitelline placenta does exist very early in human development, but it is quickly replaced. When an exam asks for a deformative human placenta, the answer is chorioallantoic. So let's talk about the second criteria, types of placenta according to the histology. This is the most conceptually important section, so let's build it carefully. Ask yourself, when maternal blood and fetal blood sit side by side in the placenta, how many tissue layers physically separate them? Because maternal and fetal blood never mix. Everything must diffuse across those layers. The fewer the layer, the more efficient the exchange. So the 6 tissue barrier. In the most complete case, there is 6 tissue barriers. 3 are contributed by fetus and 3 are contributed by the mother. Contribution by the fetus from the inside out. The first one would be endothelium of the fetal blood vessel, fetal connective tissue, and then chorionic epithelial cell of the fetus, which is also known as the trophoblast. And contribution by the mother from outside to in is uterine epithelium, uterine connective tissue, and endothelium of the maternal blood vessel. Now the key idea, based upon the relationship of the embryonic villi with the uterine wall, 5 types of placenta are seen, and the logic of subsequent is beautifully simple. In each name, the first half tells you what the fetus tissue is touching, and the second half is almost always chorial. The fetal chorion, so epitheliochorial, means maternal epithelial touching fetal chorion. So let's talk about the first one, epitheliochorial placenta. Here the embryonic trophoblast and the uterine epithelium remain in close contact with each other but retain their original layers, meaning neither embryonic nor maternal tissue is damaged. This is pure apposition, no invasion at all. All 6 barriers are intact, so exchange is least effective. These are seen in horses and pigs. The next one is Syndesmochorial placenta. Now invasion begins. The uterine epithelium is ruptured and the chorionic epithelium comes out into contact with the connective tissue of uterine wall. 1 maternal layer gone, now 5 remains. These are seen in sheep and cattle. The next one we will talk about is Endotheliochorial placenta. Deeper still, not only the uterine epithelial but also the uterine connective tissue is eroded, so that the chorionic epithelial comes into contact with the endothelium of the maternal blood vessel. 2 maternal layers are gone, now 4 are remaining. And these are seen in dogs and cats, mostly carnivores. The next we'll talk about is Haemochorial placenta. Now the descriptive step is the endothelium of the maternal blood vessel disappears and the chorionic villi comes into direct contact with maternal blood. Think about what that means physically. The villi are now bathed directly in pool of the maternal blood. No maternal tissue is in between at all. All 3 maternal barriers are gone, leaving only 3 fetal ones. And these are seen in monkeys, apes, and some insectivores. And this also includes humans as well. The next one we'll talk about is Haemoendothelial placenta. The most extreme and most efficient type. Here all 3 maternal tissues and 2 of the fetal tissue, the chorionic epithelium and chorionic connective tissue are completely eroded. The fetal blood vessel dips directly into the blood lunae, which is blood-filled space of the uterus. So the result would be the number of tissue barriers between the maternal and fetal blood streams is reduced to just 1. Only the endothelium of the fetal blood vessel is there. The result is simple. The number of tissues barrier between the maternal and fetal blood stream is reduced to just 1. Only the epithelium of the fetal blood vessel is there. And these are seen in rats, guinea pigs, and rabbits. The next criteria we are gonna see is types of placenta according to the fate of the uterine endothelium at the time of parturition. Now a completely different question. At the time of birth, what happens to the mother's uterine lining? Does it comes away with the placenta or not? On the basis of these, there are 3 types. And notice, this follows directly from what we just learned. A placenta never invades can detach cleanly. A placenta that invades deeply cannot. Non-deciduous placenta, which is also known as semi-placenta. Here's the contact between the fetal and uterine wall is not intimate. So no part of the uterine layer is shed after the birth and no bleeding takes place. The villi simply slip out of the crypt like fingers out of the glove. These are seen in horses, zebras, and logically there are the same animals with epitheliochorial placenta, which is non-invasive placenta. The next one is deciduous placenta. Here the union of maternal and fetal tissue is so intimate that at the time of childbirth, a variable amount of maternal tissue is pulled out, resulting in the shedding of the blood. These are seen in rabbit, humans, and monkey. This is why human birth involves delivery of the afterbirth and postpartum bleeding. The deeply invasive haemochorial placenta takes maternal endothelium with it. So the next type we are going to study is contra-deciduous placenta. The rarest and strangest type. The connection between the fetal villi and the uterine crypt becomes so intimate that at the time of birth, a portion of allantoic placenta is left behind and is reabsorbed into the uterine wall. So instead of mother losing tissue to the fetus leaves tissue behind in the mother and she digests it and reabsorbs it. And these are seen in talpa, which is a mole, and perimels, which is a bandicoot. So the next criteria we are going to discuss is types of placenta according to distribution of villi. The final criteria is most visual and the easiest marks in the exam if you learn the shapes. So the question is, how are the chorionic villi arranged over the surface of the chorion? There are 6 patterns. First one is diffuse. So villi remains scattered all over the surface of the chorion. The entire chorion participate in exchange, and these are seen in pigs and horses. The next one is cotyledonary. Villi are arranged in separate tufts or patches called cotyledons. Each cotyledons docks with the maternal cranicles to form a functional unit. And these are seen in goats, cows, sheep, mostly the ruminants. The third type is intermediate. Exactly what the name says. Villi are arranged in cotyledons as well as scattered, a hybrid of first two. And these are seen in camel and giraffe. The next one is zonary. Villi are arranged in a defined bands, grids or zones around the embryo. And these are seen in carnivores like cats and dogs. The next one is discoidal. Villi are arranged in small disc area, and these are seen in rats, rabbit and bat. The sixth one is metadiscoid. The key here is that it is developmental, a change over time. Villi first occurs all over but are later restricted to a small disc-like area. The disc may be monodiscoidal, which has one disc, or bidiscoidal, which has two discs. And these are seen in human, monkey and apes. Human are monodiscoidal, some monkeys are bidiscoidal. And now quickly let's just discuss the function of placenta. Finally, what does this organ actually do? There are various functions. First one is nutrition. All the nutritive elements from the maternal blood pass into the fetus through the placenta. Glucose, amino acid, fatty acid, vitamins, minerals. The placenta is fetus's digestive system. Second one is respiration. Through the placenta, oxygen passes from maternal blood to fetal blood and carbon dioxide passes in the reverse direction. The placenta is the fetus's lungs. The third one is excretion. The fetal excretory product diffuses into maternal blood through the placenta and excreted by the mother. The placenta is fetus's kidney also. The fourth one is storage. The placenta stores fat, glycogen and other reserves, resulting glucose to the fetus as needed. The placenta is, in this sense, the fetus's liver as well. The fifth one is endocrine function. It acts as an endocrine gland which secretes hormones like estrogen, progesterone and human chorionic gonadotropin, which is HCG. which help in maintaining pregnancy, supporting fetus growth, and driving metabolic changes within the blood of the mother. One note on HCG is that it maintains the corpus luteum in early pregnancy. So progesterone keeps flowing and the endometrium isn't shed. It is also the hormone detected by home pregnancy test. Let's quickly recap. So the first criteria we read was by fetal membrane. We've read yolk sac, chorionic and allantoic. And by histology, epitheliochorial, syndesmochorial, endotheliochorial, haemochorial, haemoendothelial, a single continuum of increasing invasion and decreasing barriers from 6 down to 1. And by the fate of endometrium, the non-deciduous, deciduous and contra-deciduous. The fourth criteria we have read was by the distribution of villi: diffuse, cotyledonary, intermediate, zonary, discoidal and metadiscoidal. And few functions of placenta, which was nutrition, respiration, excretion, storage and the endocrine function. So one organ built by two individuals doing the work for 4 organ systems and then discarded. That's the placenta. If this made the classification finally click, that's exactly what our channel is here for. So keep learning, keep achieving. I'll see you in the next video. Thank you.